welcome to those who are joining. Thank you so much for joining us for CellarisBio's monthly webinar. And we'll get started in just a little bit, 1 minute. Greetings to those who are already joining. Welcome, everyone. All right, well, welcome to this month's webinar hosted by CellarisBio. Today's fascinating presentation will feature two expert speakers on the topic of novel druggable targets. We have Dr. Stephen Horrigan and Dr. Elmar Nurmemmedov. Just a little bit of background of both of our eligible and very knowledgeable speakers today. First, we have Steve Horrigan, PhD in biology from Syracuse University. Steve has extensive work experience in the field of research and scientific leadership. Steve began his career in the year 2000 as the Vice President of Research at Avalon Pharmaceuticals, where he worked until 2009. In 2010, he became the Chief Scientific Officer at Noble Life Sciences and held that position until 2011. Steve then moved to Iterion Therapeutics, where he currently serves as the Chief Scientific Officer. Thank you for joining us today, Steve. Great to hear about your history there. Now, Elmar Nurmemmedov, also PhD and MBA. A little bit about Elmar's background. Elmar is the co-founder at CellarisBio. He holds a PhD in molecular biophysics. Trained at Harvard Medical School and Scripps Research Institute, later principal investigator at St. John's Cancer Institute, his research focused on in vitro pharmacology and early drug discovery. Elmar holds patents for small molecule therapeutics and is inventor of several novel drug discovery technologies. So round of applause for our speakers today. And just a little bit of housekeeping before we get started. The webinar will last about 45 minutes. And also you'll notice the Q&A button at the bottom of your screen. You can feel free to use that throughout the webinar and punch in any questions that may come up. We'll dedicate about 10 minutes at the end for our professionals to spend some time answering those questions. Now we are anxious to hear from our first speaker, Steve Horrigan. Welcome, Steve. Go ahead, it's all
Thank you, Brooke.
you.
Thank you very much. Thanks everyone for joining today. So I'm going to talk a little bit about a program that we're working on at Iterion Therapeutics, which is a small molecule that targets a new protein in the Wnt beta-catenin pathway called TBL1. And this is just a short introduction to the program. So the small molecule I'm going to talk about is called Tegavivint. This is now a clinical stage compound, and it is a binder to a protein called TBL1, which I'll talk about extensively in a few minutes around this drug discovery efforts. Functionally, it's an inhibitor of the Wnt beta-catenin signaling pathway. And it has a therapeutic potential for a broad range of tumor types. Wnt beta-catenin is one of the predominant mutated genes in cancer, as beta-catenin itself is an oncogene. It is activated and mutated in a variety of different kinds of cancer, most notably in colorectal cancer, where there's mutations and activation in essentially all colorectal cancer. In a disease we'll talk about, hepatocellular carcinoma, where a significant number, up to about 40 or 50% of tumors have activation and mutation in this pathway. As I mentioned, Tegavivint is currently in clinical trials in hepatocellular carcinoma, and also with a set of our very, very good investigator-initiated studies at several institutions, which include non-small cell lung cancer, AML, and in collaboration with NIH COG, Pediatric Solid Tumor Trial. This is just a little bit on the background of beta-catenin. A little delay on my screen change. And just to point out, this is a pathway discovered about 20 or 30 years ago in cancer that's been the target of multiple drug discovery efforts. There are a lot of technical problems with drugging this pathway. The most formidable one is that beta-catenin itself is not conventionally druggable. It's not an enzyme or a receptor. It is a long, unstructured protein that has repeats of various forms across it. And conventional small molecule targeting of beta-catenin itself has been difficult. Targeting other parts of the pathway have been tried in various cases, and that has led to either toxicity or lack of activity because of the plasticity of the beta-catenin pathway. There have been recent efforts by us and several other companies to actually target beta-catenin pathway using novel interacting proteins. The most prominent ones were the tankerase inhibitors, which multiple companies had tested and had in clinical trials. That those have some downsides in that tankerase actually has on-target toxicity off the beta-catenin pathway, which is limited in its clinical development. In our case, we have a compound that was discovered to a novel druggable target in the beta-catenin pathway, which we'll discuss in a moment. And this is just a little bit around how the compound was found, how TBL1 was discovered, and why we were able to identify TBL1 as a druggable target. And it was previously known to be a regulator of beta-catenin, but it was not known to be a binder of small molecules. And this is a novel screen that was a chemical genomic screen, so based on a cancer cell that had a mutation to activate beta-catenin, and the system used a set of gene expression targets, which were genes which were specifically regulated by the transcriptional activity of beta-catenin. The logic behind the screen was that transcriptional activity, the ability of beta-catenin to act as a transcriptional regulator, is its oncogenic activity. So inhibiting that as directly as possible was the intent of the screen. And I think in the experience of the last 10 or 20 years, that was the correct strategy, and that inhibiting the Wnt beta-catenin pathway upstream and on other targets in the non-transcriptional activity leads to either toxicities or lack of activity. The weakness of this kind of screening, of course, is that it's target agnostic, in that even though we had a quite potent compound that was a quite good inhibitor of beta-catenin pathway, and it had all the characteristics of it, the target of the compound, the protein target of the compound, was not known. Through chemical biology and a lot of binding, we discovered that TBL1 was actually the target of TBL1, binds directly to TBL1, and TBL1 itself was a known regulator of the beta-catenin pathway. This is just a little cartoon about how it does that. So TBL1 binds to beta-catenin in the nucleus and acts as a recruiter to the transcriptional of the transcriptional complex to the relevant target genes for beta-catenin. And so now we're targeting directly at the transcriptional complex and not of any of the other functions of beta-catenin, which are necessary for normal cellular function. The right-hand side picture in this case is just to remind me there is now a crystal structure of TBL1, and I'll show you some work we've done on characterizing interaction of TBL1 with TBL1 in its structure, the protein structure. TBL1 itself is an oncogene. It's actually known as a regulator of multiple oncogenic pathways, most prominently nuclear beta-catenin. It has both mutations and translocations and amplifications in cancer, and has a function in and of itself that and it is a regulator of the protein stability of oncogenic transcription factors. And so in our case, we were able to discover a compound that inhibited its beta-catenin activity. As you can see on the left-hand side, and this will become relevant later, TBL1 is a quite complex protein structure. It's a tetramer of TBL1 and multiple genes. So there are 3 separate genes for TBL1 family members on 3 different chromosomal locations. And each of these can assemble into homo and heterodimers and homo and heterotetramers. So the typical, simple protein target that you can analyze in enzyme activity is not this program. And to be relevant to this thing, most of the oncogenic targets in the world are complicated and complicated protein-protein interactions. The activity of TBL1 is associated with very poor prognosis in multiple tumor types, mostly because it's associated with invasion and metastasis, just like beta-catenin is. And there's a correlation between TBL1 levels, beta-catenin activity, and the ability of beta-catenin to transcriptionally regulate oncogenic function. This is the way, a short cartoon of how Tegative event works. On the left-hand side is the normal Wnt beta-catenin pathway, where beta-catenin binds to TBL1. The nuclear degradation of pathway, which degrades beta-catenin to control its function through the CO1 complex, is blocked in this case, and beta-catenin and TBL1 co-recruit each other at high affinity to TCF binding sites or to beta-catenin binding sites to set off a series of oncogenic events in the nucleus. One of the key components of this is that this interaction and target is below where the majority of mutations happen in cancer. So mutations in beta-catenin or in APC or in axin that activate the Wnt beta-catenin pathway are above it. So Tegative event actually is functionally active in all those settings, since it blocks the function of beta-catenin, not in those degradation complexes, but below it. On the right-hand side, you see the cartoon of the way Tegative event works. It binds directly to TBL1 in the pocket that beta-catenin binds into. Beta-catenin is released in the nucleus. That allows it to be degraded by the CO1 degradation complex in the nuclear function. That turns off the oncogenic activity through the transcriptional function. It does not cause degradation of the membrane-bound beta-catenin, which is very important for cell-cell interaction, and the non-canonical pathways, which are important for bone function and other normal physiological functions. This is just a little bit of data around how the compound works. This work was done by Rafi Soldi and Sunil Sharma's group, both in collaboration with Dr. Sharma's laboratory and at Iterion Therapeutics, and this was published over the last couple of years. Just to show that beta-catenin is degraded in the nucleus. This actually is experiment done by Daniel Braggio at Ohio State University in desmoid tumors, which is what our initial clinical trial was done in, showing very nice activity in this very hard to treat tumor type. Right-hand side is just is showing inhibition of beta-catenin transcriptional activity, and most relevant, I think, is the binding side on the right-hand side, where the bottom panel is a proximity ligation assay showing that we can disrupt beta-catenin and TBL1 activity. The pink signal is where both those proteins are bound together, and after compound treatment, the beta-catenin and TBL1 are not bound together, and you can show this in non-cellular systems also. These are nice functional assays, but they're very cumbersome to do, and they're very difficult to generate potency data off them for determining both tegovince activity and new compounds we might want to interrogate. That's one of the significant problems with a target like this, is that generating good quantitative data is difficult on assays where there's not a traditional enzymatic or proteolytic event. To look at a little more of the function, TBL1 itself is a nuclear protein. These are the two major analogs that are found in both cancer and normal cells. The third gene is TBL1Y is a Y-linked gene and that's why it's named that way. It's expressed very little in adult tissues and in cancer. These are the two predominant isoforms expressed everywhere. And you see this is an immunoprecipitation to show that if you treat with tegovince over the first 6 to 12 hours, that disrupts interaction of TBL1 with beta-catenin, but it doesn't disrupt its other complex family members that we've looked at. The nice thing about this system now is we can generate a truncated version of TBL1. TBL1 has two functional domains: an N-terminal domain, which is responsible for beta-catenin binding, and the C-terminal domain, which is responsible for bringing in transcription complex. And our work and work by others have shown that this N-terminal domain is sufficient for the beta-catenin and the tetramer complex formation. This is just to show that part of this interaction, you can see this is a computational model in the middle of TBL1 using the published crystal structure of TBL1 of this tetramerization domain with tegovince bound into the pocket. This pocket has been described by others as the beta-catenin binding site, in allowing beta-catenin to not interact with TBL1. TBL1 itself is not degraded by tegovince, so it recycles. It's a necessary protein. Deletion and knockout of TBL1 and TBLXR1 are lethal to cells. So one of them is allowed and both of them aren't. This actually presents another problem or challenge of the program, in that there are two very, very similar proteins that bind the small molecule compound. So looking at, for instance, CRISPR screening technology, looking for activity of TBL1 wouldn't work, because you'd have to knock out both genes simultaneously to see it. And that allows this complicated system to assemble into what is a complicated transcriptional complex. This is just where we are in the clinical trials of tegovince from its function. Like I said, it is currently in a phase 1/2 clinical trial in hepatocellular carcinoma that is ongoing right now and enrolling. It is also currently in investigator-initiated trials, both for acute myeloid leukemia. Kalpala's laboratory at MD Anderson has done a lot of work with tegovince and AML to show that it has both anti-tumor activity in AML and can target AML stem cells. There is a study in pediatric solid tumors run with COG, which is Children's Oncology Group at NCI. This is really based on our collaborator Jason Eustein's work in osteosarcoma, showing that beta-catenin is activated in metastatic osteosarcoma and causes a lot of biological functions which are detrimental to treating those aggressive tumors. And tegovince is very active in preclinical models in that case, and now there's an ongoing study in that. For non-small cell lung cancer, this is done at Ohio State University with David Carbone and Regan Memmott. this is a study in combination based on their work that shows that EGFR inhibitors, which are excellent drugs and work very well, but like most kinase inhibitors, essentially everyone relapses off those drugs and progresses. That if you treat non-small cell lung cancer patients with EGFR inhibitors in combination with WNT inhibitors, you can reduce these persister cells, or these cells that are the relapse generating cells. This is a very exciting study that they're doing now, and is rolling very well. And then the last one is Lapo Olinari in B-cell lymphoma has published some excellent data on tegavimab's activity in B-cell lymphoma. TBL1 is a known oncogene in lymphoma. It's mutated, it's translocated, it's activated. It has a special activity in immortalizing and causing oncogenic transformation in the lymphoma cells. And he's started a clinical trial treating lymphoma patients, particularly double hit, triple hit, meaning both beta-catenin, MYC amplification, and VCL anti-apoptotic mutations, which are very difficult patients to treat. So what are the challenges for us? One is TBL1 is a very complicated protein structure. So doing simple assays and simple drug discovery things was difficult. The complex itself, because it's a heteron homer dimer, forms multiple binding sites per complex, and these complexes are switchable so that as TBL1 is modified, for instance, by sumylation, it exchanges beta-catenin for NCOR complex in this case. So it's quite difficult to do conventional assays to do it. It's also regulated by phosphorylation to do other functions. So you need to, in the case of, for instance, generating protein in a bacterial system that's a simple, protein you can generate for kinase activity is actually quite difficult because the protein is not modified and assembled correctly as its functional part. And having multiple genes that encode for very similar functions has been difficult. And this is part of the problem. I think on the right hand side, basically is from a publication by Choi et al. Showing how this system runs in a continuous recycling event where TBL1 complex binds to NCOR SMART, is modified by simulation, and that simulation is controlled by Wnt signaling. That turns it into a beta-catenin binder. Once it's released from the Wnt signal, it releases beta-catenin and it goes back to the NCOR SMART binding. So we need a smart system for binding assays that are done in a competent cellular system, but don't cause the complex interactions to destroy the assay. These are the three genes, and you can see that actually they're essentially identical over the N-terminal binding region, up to 95% identical in amino acid function and sequence. And that's where beta-catenin binds and where tegavimab binds. And so what are we doing to overcome that? And we actually have produced TBL1 and TBLXR1 in bacterial systems for binding studies. It's a difficult process to do that. Proteins need to be refolded and modified to be able to bind, and they only assemble partially. We have worked with doing a computational model. There is, like I mentioned, there's a crystal structure of TBL1 that is known. We've been working with Conifer Point Pharmaceuticals, which is a computational chemistry molecular design group, very successfully to identify analogs of tegavimab, which have increased pharmaceutical properties and bind to TBL1. And now we're moving those on to direct binding assays for interactions to TBL1. Our partner Viva Pharmaceuticals has developed the ASNS assay, which is able to show binding directly to TBL1 in the bacterial purified proteins system. And now our collaboration with CellarisBio that has been ongoing for several years now, really to show the interaction of these proteins in integrative event with the complex of TBL1, where it's assembled into a rational complex and it's in its natural setting. So now I'm going to pass on the rest of the talk on to Elmar, and he'll describe both the system for analyzing binding of small molecules and some of our data we've generated together with integrative event.
Thank you so much, Steve. We had a problem with the slide transition. So at CellarisBio, we are all about cellular target engagement. And what we do is we like to take challenging drug targets like TBL1 and really establish assay systems so we can de-risk and accelerate early drug discovery, especially the MTA cycle. As you already have heard, TBL1 is a challenging drug target for various reasons. It has a few of these hallmarks. There is a lack of hydrophobic pocket. There is a lack of active site per se. And TBL1 is involved in protein-protein interactions. Or its active site, if it was available, would be conserved among other family members like the TBL1X and the TBLY. So essentially, it has a few of those hallmarks, making TBL1 a challenging drug target. This is why looking at this target within its natural context, within the cell, is very important, which actually conserves its function and structure that makes it more disease relevant. So at CellarisBio, we have assembled this enabling workflow for early drug discovery, and we have employed it for the purpose of TBL1 program. So the first step we do is we select the target as our our assay system. In this case, TBL1, TBLXR1, and TBL1Y are the three isoforms of TBL family that we have assembled together, and we have generated assay systems for that. So we engineer the microtag reporter cells by introducing the 15 amino acid tag to the N or the C terminus of the target. And essentially, we can do it in a transient expression using as simple as hex cells, or we can really lentivirally transduce cancer-relevant cells and make stable reporter cells, as we have done in the case of TBL1. After that, essentially, it becomes a smooth sail. We can engage now, we can use the engineered stable reporter cells for heat discovery if that was necessary. And in the case for TBL1 program, we can do heat validation. Essentially, we can easily interrogate the three targets, three isoforms against the main compounds, main candidates in this program against the target in the cell, and this can push lead optimization. This can essentially transition from hit to lead generation and de-risk your medicinal chemistry and make the entire program work much more predictably. And most recently, we have added the real-time analysis capability that allows us to interrogate the in-cell kinetics of those interactions. Essentially, this is all to say that we can de-risk and accelerate preclinical studies for challenging and complex targets like TBL1. And once the system is assembled, now we can feed more candidates into the system and make the cycle go much faster, smoother, in order to generate better analogs so that we can push into preclinical and hopefully clinical programs. So the way our microtag cell target engagement system works is by assembling a reporter system in the first place. So you will define your target in the first place. It could be a nuclear target, like in the case of TBL1. It could be a cytoplasmic target, or it could be a membrane protein. The first step is clonally attach a 15 amino acid tag to the N or the C terminus of that target, making now a reporter cell for that specific target or family of targets. You will treat the cells with your ligands either in a pooled fashion or in an individual fashion. Temperature interrogation is important to really bring your target in the cell to a point of denaturation. And the foundation of this technology, the principle is that when a ligand, a drug candidate, binds to the target inside the cell, it's supposed to biophysically stabilize the target and rescue it from the temperature-induced denaturation event. And when we are ready to detect a signal, essentially we would add tag binding protein and a FRET substrate to the reaction. So what happens is that the 15 amino acid tag that sits with the target and the tag binding protein added to the solution now will complement into an active enzyme. It will cleave the FRET substrate and release the fluorescence signal, which is a dose-dependent event. And the amount of fluorescence signal is directly proportional to how much your target was stabilized by your ligand inside the cell. Now, this is a very simple system. It can go into conventional plate readers to measure the endpoint fluorescence. This is a conventional way of measuring the signal. Most recently, we have upgraded the system to be read in real-time machines like this one here, where we can read the entire flow of the reaction from time 0 all the way to time X, giving you the ability to look at the stoichiometry and phase transitions of cellular target engagement. And you can also go into real-time live cell imaging systems to really understand in more detail what happens to the target as a result of this engagement. Is there accumulation with the target? Is there degradation? Essentially giving you a little more mechanistic insight. So for TBL1 program, we have started with panel A. Essential, this is our quality control. We have generated those several DNA constructs for each of the isoforms. And the one thing we do, we want to put those back into the cells of interest, and using an anti-microtag antibody, look at the expression levels. We want to see pretty much very clean bands. We want to see single band preferably, and we want to see that there are no degradation products. We can, sometimes we will see oligomerization states or some kind of post-translational modifications, and this will give us an insight on how the assay system will progress from here. Panel B is pretty much a continuation of the quality control, where we look at the amount of fluorescence signal generated by these microtag targets with that 15 amino acid tag attached to them. And we can see that against the untransfected background, the level of fluorescence is sufficient for us to really confidently move into testing these compounds, testing the analogs, testing these targets against the candidates in the cell system that we have developed. Panel C is the thermal profiles of these various isoforms of TBL1. We want to pretty much establish temperatures at which these targets would be susceptible to denaturation, and they would interrogate the small molecules against these targets at these specific temperatures. And as you can see, TBLXR1 melts at 56 degrees, L1X at 52 degrees, and L1Y denatures at 54 degrees. And we have actin-1 as a control as well. Basically, we can interrogate all of these targets at pretty much identical temperatures. And there is a convenience there. So we went ahead and we tested our main compound tegavivint against the TBLXR1, and we have pretty much obtained an EC50 of 1.2 micromolar. So you can see there is a target stabilization profile for tegavivint. It starts from low temperature, and in a dose-dependent manner, it will transition to a higher temperature and it will basically stabilize. So the flexing point here is the EC50. This is pretty much the dose of the ligand tegavivint at which half of the population of the target inside the cell has been stabilized. And this interrogation happens at that particular temperature at which the target is vulnerable. We have went ahead and we have tested TBL1X to the same ligand, tegavivint, and we have measured identical EC50 micromolar, suggesting that tegavivint is equally strongly interacts with the two isoforms as we have previously predicted. And this sets the base for our expectations for the future of this program. Additionally, what we have done is we have tested other inactive analogs of tegavivint against the main two targets now. So here on the top left panel, you will see an inactive analog engaging with TBLXR1, and we see that there is no profile. Essentially, there is very weak engagement or there is no engagement at all. And we have also tested the inactive compound against TBL1X. And again, we see that there is no profile of target stabilization, giving us confidence that there is a selectivity against active compounds. And this assay system now has been built to really go faster in a scalable way, so we can onboard newer compounds and push forward the medicinal chemistry in a more predictable way. And we have also tested actin against tegavivint, and we can see that neither the tegavivint itself or its analogs are interacting with actin. So again, there is a confidence in selectivity. There's a confidence in the assay system that we have built that we can easily and reliably scale. And really, we are building this system so that we can de-risk and we can accelerate the MTA cycle. And now, depending on the family of the target protein and depending on rationale of drug discovery biology of that target, various components can come into the picture during the DMTA cycle. For TBL1 program, it's important to look at the structural basic design aspects of the target versus the new ligands. It's important to look at the medicinal qualities of these various analogs. And again, we want to tie all of these components of the DMTA cycle through cellular target engagement. Steve, would you like to add anything onto this slide here?
Sure. Thanks, Elmar. I will just mention that, this allows on the left-hand side for us to enter new compounds and new structures by multiple different techniques and technologies, whether that's structure-based design, conventional screening, or outside, and then rapidly test those in a relevant cellular system in a label-free method. So because all the compound structures are different, we don't have to label compounds, we don't have to do any chemistry to put handles on them, we don't have to do anything but take the compound, treat the cell lines we've already generated and we already have, in hand, and test these new compounds for comparative binding to, a tegavivint or other analogs.
Thanks, Steve. So the upgraded microtag platform now offers a wider spectrum of possibilities. Target interrogation has been one of the key avenues for us. Essentially, we can feed into our platform various target families. We like to work with challenging drug targets, transcription families, regulators, and we can push them through our platform, apply the discover, validate, and now analyze components of our platform, and essentially we can generate initial set of data to really understand whether or not targets could be tractable for drug discovery purposes in our system. So initial data will show you pretty much the thermal melting profile for a target, its potential target stabilization profile with existing tool compounds, or perhaps some promiscuous compounds. And this initial data will be able to, with confidence, guide the next steps in that drug discovery if those targets are chosen for future programs. We have also upgraded the system with the real-time cell target engagement possibility. So this newer system bypasses some of the previous steps that we included in the conventional cell target engagement screen. Essentially, Once the microtag expressing reporter cells have been established, you can now treat those cells with your ligands, with your drug candidates in dose-dependent manner, and the reaction plates can go into a real-time machine, and we can basically multiplex various temperatures against various doses of your test compounds, bypassing some of the previous steps like thermal profiling, like understanding the temperatures at which you want to interrogate the small molecules. So this very quick and scalable new method will allow you to compare your target's behavior in the system and to assess its stabilization or destabilization profile against multiple compounds in a same setting. So here on the right side panel, you will see the profile for a DMSO. This is a red line. And as you can see that the amount of fluorescence signal will wind down and plateau as we increase the temperature, as we go from lower temperature, say 40 degrees towards 70 degrees. And you will see that the fluorescence signal will wind down. When we test that with a tool compound, a positive control, we will see that there is a point at which the DMSO will essentially separate from the test compound, suggesting that there is a thermal melting event happening here, beyond which the test compound will stabilize the target to a point that you will get more and more fluorescence signals. So the area between the curves here is the magnitude of stabilization of the target by the ligand you're testing. So this is a new, very scalable and very sensitive way to do cellular target engagement. I just want to wrap up this by saying that conventional challenging drug targets can be the source of first-in-class programs, like in the case of TBL1 program. The conventional path will take you for 5 to 7 years without clarity in cellular target engagement, because you'll have to do additional validation and screening, and you will have to basically tie the various components of this multi-parameter optimization effort, eventually leading you a drug candidate. Will take more time and resources. Using the cellular target engagement platform, now you can de-risk and accelerate early drug discovery, tying those loose parts together in a much efficient way. The programs will essentially evolve in a much faster pace within 1 and 2 years, because now there is a clarity in cellular target engagement. You can bypass further validation. You can bypass certain conventional biophysical tests, and you can go from hit optimization to candidate generation in a much efficient pace. I will stop here, and Brooke, back to you.
Thank you. Thank you, Steve and Elmar, both of you, for your expertise on these interesting topics. So we'll open up our Q&A session now. And as a reminder, you can still punch in your questions there in the little Q&A box. Feel free to type in any questions you may have. And let's get our experts up here with me. Hold on a second. Okay, there we go. So that you can help us answer some of these questions. Got 2 of us up here, and here comes Steve. Fantastic. Okay, so one question that will be really helpful for everyone. Does microtag cell target engagement system also provide a functional readout of the target? Could either of you help us to answer that?
I could take that. So essentially, cellular target engagement is an assay system that measures the biophysical events happening directly inside a cell. So essentially, there is a target that you have defined, and there are drug candidates that you are testing against the target. And the system is designed to measure those early biophysical events happening in the cell. So, and the EC50 or the in-cell KD, the affinity, are the measures of that, of the strength of that engagement directly in the cell. Is there a biological relevance? Not directly. The system is agnostic to whether your target is an active site, carrier, or basically a challenging target without an active site. regardless of the active site, regardless of the activity state of the target inside the cell, the system will measure whether or not there's a biophysical event between the target and the ligand. In the best case, you may want to complement your cellular target engagement data, your cellular target engagement system, with biological readout, suggesting that because of this biophysical interaction between the target and the ligand, now you have this biological readout. It could be cell death, it could be inactivation of the target, it could be degradation of the target. So depending on the therapeutic modality, you may need to bring in some biological readout to really complete the system and complete the answer that you're going after.
Thank you for that, Elmar. Okay, I think we have time for one more quick question. How does microtag compare to SPR or related technologies for TBL1 potency assays? Do you want to help us with that,
Thanks.
Steve?
Yes, absolutely. So for TBL1, SPR assays actually perform quite poorly, and that's been, it's one of the problem with this program and with many of the transcription factor programs, in that because it requires assembly of a protein complex and the compounds that bind into these hydrophobic pockets for protein-protein interactions are also hydrophobic compounds. Technically, SPR does not work well. And so you need a system like the microtag that's kind of in a more relevant biological system where you can assemble protein complexes and where you can treat it in a relevant mix rather than buffer. And now it's actually in the protein mix of a nuclear extract or a cellular extract.
Fantastic. Thank you. Well, I think our time is up for today. Thank you all for joining us. You've been a fantastic panel and audience. And as you can see on the screen, you can get in touch with us and ask any other questions, emailing hi@celarisbio.com. You can also follow us on LinkedIn and our website, CelarisBio.com, where you will find a recording of this webinar as well as previous webinars. Also, look out for future webinars by CelarisBio, and we hope to see you again soon. Have a great day, everyone.